Hello, students, and welcome to Cell and Molecular Biology. This is spring 2020. This is Professor Blaine Moore coming to you live from my basement in Kalamazoo. Did not expect to be interacting with you this way, but we're in different times, and different times require different solutions, so here we are. I'm going to do my best to make this course as engaging as possible, and I wish you all the best of health and luck as we move forward through the term. Our first lecture, Lecture 1, is going to be divided into two parts. Part 1 is going to be essentially this part of my outline, beginning with a brief discussion setting a foundation for what we're going to learn, a little discussion on tips for success in the course, just unsolicited advice that I'll give you, and then finally an introduction to the design of the course from a topics perspective, a syllabus and expectations perspective, and a lab perspective. Part 2 of this first lecture will be the actual Lecture 1 material, and that will be posted as a separate link. Let's begin with the discussion. And what I mean to do here is to convince you, hopefully, that we all share a scientific foundation upon which we can build with our new material that we'll be learning in the Cell and Molec course. So let's begin by imagining the source of all life here on Earth, and that's the sun. The sun's about 93 million miles away, and light released from the sun travels over about 8 minutes to reach us here on Earth, and it gets absorbed by particular organelles within the primary producers, the plants. So inside this organelle, the chloroplast, there are particular photopigment molecules like chlorophyll, which can absorb that photon of light. And there's a water splitting enzyme that's capable of splitting water to elevate some electrons to a higher energy state that's catalyzed by light. And then those electrons fall down their energy gradient. And all along, we have the movement of protons, and the movement of protons are used through this electron transport chain to produce particular types of energy currency, NADPH and a little bit of ATP, for example. Now, what I've just described are the light reactions. As you might guess, they depend on light. That's how they get their name. Now, once this energy currency is produced, we move into the dark reactions. And in the dark reactions, carbon is fixed. It's fixed by an enzyme Rubisco, perhaps the most abundant enzyme on Earth. And that energy currency from the light-dependent part of the photosynthesis reaction scheme is utilized to drive the reduction of that fixed CO2. And what we end up with at the end of the Calvin cycle, or the dark reactions, depending on how you prefer to refer to them, what we end up with is sugar. Now, let's follow that energy. Remember, this all originated with the sun. Let's follow that energy through an ecosystem and imagine that a primary consumer eats the plant that contains this stored sugar that it's used photons of light to essentially build. Well, now that material, that sugar, is in the digestive system of the primary consumer, and digestive enzymes will break that sugar down. It will make its way into the blood, and then the sugar will be carried through the blood and make it to the outside of cells, the extracellular space. Now, a sugar molecule is large and polar, and it's unable to slip across the plasma membrane of an animal cell. It needs a carrier, it needs a protein to facilitate its movement across the membrane. And of course, it's the glucose transporter that does this. Later on, we'll be covering this mechanism, but suffice it to say, the glucose transporter brings the glucose, the sugar, from the outside in, and there in the cytoplasm, we begin a series of biochemical reactions called glycolysis. And in glycolysis, what we're doing in aerobic conditions is we're starting with glucose, and through a series of chemical changes, ending up with a chemical called pyruvate. Remember that in anaerobic conditions, lactic acid is the main product of glycolysis. But since we're dealing with aerobic conditions, we'll assume it's pyruvate, and then we'll think about where that pyruvate goes. Now, inside the cell, remember that glycolysis really happens in the cytoplasm. Pyruvate needs to move into an organelle to be acted on to create energy currency for the animal cell. And you probably know where it's going, but it's going to end up, if you don't know, in mitochondria. And that pyruvate's going to move across the outer membrane of the mitochondria, across the intermembranous space, and eventually into the mitochondrial matrix. And there, that pyruvate's going to be decarboxylated, combined with acetyl-CoA, and the resulting product will enter the Krebs cycle. And the Krebs cycle is kind of analogous to what we talked about with the Calvin cycle that happens in plants. We're going to start with this material, and we're going to end up with essentially energy currency. Now, this energy currency is the form of NADH and FADH2. Now, these essentially are the electron donors for the electron transport chain that's embedded in the inner membrane of the mitochondrion. And you'll probably remember that electrons get stripped off of these electron donors and essentially handed through different complexes. Protons get pumped, a gradient builds up in the intermembranous space, and then it flows through ATP synthase. And ATP synthase is important because it produces ATP, which is the ultimate energy currency of the cell. Now I want you to realize, and I think this is fascinating, and really frankly every time I think about this it blows my mind, the high energy bonds within ATP essentially originate as a photon of light from the sun. And of course that energy has been converted and manipulated through multiple forms, but essentially this energy currency is sun derived. It's pretty cool. So if we were sitting together in the classroom, I would ask you all to give me some examples of what ATP can do. Why is it such useful energy currency? For example, we're not sitting together in a room, unfortunately, so I'll just give you a few examples. So one thing that you'll probably remember from other courses you've taken is that cells need to maintain a certain membrane potential, and membrane potential is a way of thinking about this is to think of it as a potential energy source that the cell can harness to use for work. The way that ATP helps to maintain membrane potential is by driving the action of pumps for particular ions, and the one you're most familiar with is the sodium-potassium ATPase. So maintaining membrane potential by driving this pump is an important role of ATP. Just one more example, and that is in the example of post-translational modification. ATP is an important molecule in that, particularly with regard to phosphorylation. And this is where an enzyme is able to catalyze the transfer of one of those phosphates from ATP to a substrate. Often enzymes use this mechanism to turn themselves on and off. And again, something we'll be talking about a lot later on. So we started with the sun, we ended up with ATP, we then looked at what ATP is doing. Everything's connected, and not surprisingly, given that this is science discipline that we're studying this term, cell and molecular biology is connected to everything else that you've learned. So when you've heard in your high school classes and your college classes about photosynthesis and cellular respiration, of course, it all is really driving the production of ATP. And what we're going to use is we're going to use that foundation as a starting point to then sort of build upon as we learn new things in this course. It's an important goal that we have in this course, and Dr. Shaven and I are going to be trying to build upon the foundation that others have really set down for us. So let's talk about course goals, and what we're going to really realize here is that the cell is the fundamental unit of life. Cell theory helps us to understand how that was conceptualized. And because the cell's the fundamental unit of life, what we learn about cells in one species are often applicable in other species. This is one of the reasons that model systems in biology are so useful. What we learn in a transgenic mouse model of human disease gives us insight into what's happening in the human because of that fundamental unit. Fundamentally, everything's the same. So that's one goal of the course. Another goal of the course is to always be thinking in terms of experiments. So one thing we'll be trying to do is to give you an empirical approach to all the questions that we'll be asking and answering. One way that we facilitate this is to really drive home the idea of techniques early and often, so that we develop sort of a toolkit of techniques, if you will, and then we can use that toolkit and apply it to all the new material that we're exposed to. So we'll be trying to give you this experimental approach as we move through the course. We'll also be covering some what we call timely topics, things like how molecular mechanisms are uncovered, how signaling within a cell is an important regulator of various processes, how microRNAs, a formerly unknown RNA species or RNA variety, help to control things like gene expression and even phenotype during development. And we'll also, towards the end of the course, talk about stem cells and their potential application clinically. So we'll hit some of these newer topics in the discipline. Another goal is to introduce you to understanding and interpreting the primary literature. So that's something we'll be hitting once we get to our later stages of the course. And as I've already indicated, we always stand upon the shoulders of giants, so to speak. That's what Newton tells us. And we will try to do something similar to that, maybe not as grandiose, but we'll try to build upon and extend concepts that you've heard in other courses. All right, so let's take a look at some nuts and bolts. Here is a sort of outline of the syllabus for the course this term. Of course, copies of all these materials will be posted on Moodle. Please read them and let us know if you have any questions. So we have the syllabus, the title of the course, obviously, your professors who are sharing the duties this term. We are running the course asynchronously, so the lecture times and the meeting times for the lab, all that stuff is flexible. Please do it at your leisure. Office hours are preferably by email for both Dr. Shaven and myself, and here you see the times that we are available. These are times that we will be available to answer email. Of course, we will answer email outside of those hours as well. You just may have to wait to get a response outside of our office hour windows. Emails are given here. And the text that we're using is we're using the 4th edition of the Alberts et al. Essential Cell Biology. If you have an earlier edition, that should be fine. You'll just need to transcribe or transpose the page numbers for the readings into your new edition. If you have a later edition, I think there is now a 5th edition of that, that will also be fine. Either one is fine, frankly, as long as you have a copy of the book. Now, I mentioned earlier that we'd talk about tips for success, and here are a couple of pieces of advice. So one is to take a look at the syllabus and look at the schedule and read before listening to the lectures. What this will do is it'll give you context for the lecture. I want you to think of the course as a large book, by way of metaphor, that's divided into 3 parts, parts 1, 2, and 3. And the first part, or the first module, contains a certain number of lectures. Think of those lectures as a chapter. And each lecture is, in our case, presented as a link on Moodle that you open up and listen to as you go through the lecture. It's very important that we always keep in mind the story of the course, the narrative that we're trying to tell, and we'll try to remind you of that throughout. So if you read beforehand, you're getting that version of the story from another source, and it gives you useful context for when you listen to our version of that story. So to the extent that you can, try to read before listening to the lecture. Then listen to the lecture and sort of watch the animations, etc., that show up on the screen. And then later on, cross-reference between the book and the lecture. Now, each of these lectures that Dr. Shaven and I will be giving will be really showing up as in two forms on Moodle. First is the actual presentation itself, and that's where you get the voiceover PowerPoint with the animation. We will also post just a PowerPoint or even just a PDF version of the slides without any narration. So you can print those, you can put them into a notebook, however you find it convenient, or just frankly look at them online, doesn't matter. But if you can cross-reference between those lecture slides and the book, that will help to shore up the knowledge that you're learning, the information that you're learning. So pre-read, listen to the lectures, and then cross-reference. That's a good way to master the material that we'll be exposing you to this term. Grading is credit, no credit for spring 2020. This was voted on by the faculty over spring break. There are no exceptions. This is the way that we're doing it. And I want you to please remember that, the most important thing is to work diligently and on time so that you're making progress in the course and working towards receiving credit. The course is really designed for all of you to receive credit in the course. And the way that we will do that is to achieve 70% or better on each assignment. If it so happens that for a particular assignment you aren't achieving 70%, you may retake the assignment and then achieve full credit. At the end of the course, as long as all the components are completed at that 70% level, you will receive credit for the course. I do not expect there to be any awardings of non-credit in this course, but of course you have to hold up your end of the bargain and approach the course professionally, turn things in on time, and work diligently. I don't expect there'll be any problems because you are case students after all. Here are the components of the course. We've already talked a little bit about these voiceover PowerPoints. These will really serve as the foundation of the course. Listen to these. They will form the basis of the other parts of the components here, the other components. Speaking of, weekly lecture discussion questions that will be posted on Fridays beginning in week 2. You will complete these on your own, asking your TA for help or asking your professors for help if you need it. The basis of these will be from the voiceover PowerPoint, so that's why it's important to listen to each of those. These will be due by Wednesday of the following week. So for example, the week 2 lecture discussion questions are due week 3 Wednesday. And they're going to be due, you'll be posting them to Moodle, and they will be due by 5 p.m. local time. So that means that you can work in your own time zone. For example, if you happen to be listening to me from California, 5 p.m. Eastern Daylight Time is 2 p.m. in California. Your assignment is not due until 5 o'clock your time. That would be 8 o'clock Eastern Daylight Time. I think you get the idea. So we're working under local time constraints for turning things in. Similarly, each week I'll be uploading a pre-lab lecture, and for weeks 2 through 9, there will be an associated lab discussion question or questions. Those are also due the following Wednesday by 5 o'clock local time. And then finally, at the very end, we'll do a brief lab report that will be due by Friday of week 10. So essentially, these are the 3 graded components of the course, and the voiceover PowerPoints serve as the sort of the main way that the material will be delivered in addition to the text. If you have questions on this, let us know. Finally, in terms of course mechanics, each student has been assigned to a TA. You've been divided into discussion groups. Yourself plus 7 other students are in a discussion group led by a single TA. Think of the TA as your academic mentor, really an extra person to help you master the material, and think of them as sort of a go-between between the professors and the student yourselves. They are an excellent group of young scholars. They are going to do an excellent job, I know, and they will help you very much in the course. Feel free to reach out to your TAs um, as often as you like through whatever mechanism they have communicated to you. Some I know are using email, some are going to be using WhatsApp, some are going to be using Zoom or some other features. But whatever the mechanism is, know that they are always there for you and they're really eager to help. I encourage you to utilize them because they're an important resource. All right. So now that we've talked about sort of the nuts and bolts of the course and we've talked about the foundation that we all share, let's talk about how we're going to build on that foundation, and let's think about how this book, by way of metaphor, is organized. So, the title of the book would be Cell and Molecular Biology. The parts of the book, there are 3 of them. Think of those parts as modules or units. Module 1, part 1, is defining the cell and its study. So defining the cell, I think that's pretty obvious. This is structure, function. How's the cell structured? What are the organelles? How does material move? But the its study may not be as clear. What I mean by that is, what's our experimental framework for understanding the cell and its structure function? What's the toolkit? What are the experiments that we have at our disposal to study this complicated piece of machinery? And I told you earlier that an empirical approach or an experimental framework is very important in the course. Well, this is where that is brought to bear, because all of the material that we'll cover we're going to think about from an experimental perspective. So that's what I mean by its study. Here are some of the topics, not exclusive or not fully inclusive, but here are some of the topics that we'll be covering in this first unit, beginning today with cell theory, the idea that the cell is the fundamental unit of life and cellular evolution. How did cells come to be, or what are our best models for that? This will include techniques and experimental model systems. Those are important to give us that experimental framework. And then finally, it's a whole lot of structure function. How's the membrane of the cell structured? How are the proteins in the cell structured? How do they work together to make the cell function normally? How does material move through the cell, this endomembrane system? What organelles drive that? What provides structure to the cell? How's it all built? And finally, how do we fuel the cell? How do enzymes work to make energy currency for the cell? So that's our first module, defining the cell and its study. Over here is a little cartoon. This gives us a sense of one of the techniques that we'll be covering, and that's the manipulation of DNA to make recombinant DNA. Here's a sort of simple example of this, just to give you a flavor for some of the topics we'll be hitting. Let's imagine that we have a plasmid. A plasmid is a sort of vector that bacteria use to share antibiotic resistance genes. Well, scientists manipulate plasmids to carry genes of interest to us. Let's imagine that this little pink region of this foreign DNA molecule is a gene of interest. We can use enzymes called restriction enzymes to cut that little pink snippet out of that larger piece of DNA. We can take that and treat it as an insert. And if we open up our plasmid by cutting it here with the same enzyme that we used over here, we get an open plasmid that we can ligate that foreign DNA into. And that creates this recombinant DNA molecule that we can then introduce into bacteria. That technique is called transformation. That's how we introduce foreign DNA into bacteria. And then every time the bacteria divide, they're going to replicate that plasmid, making many, many copies of it. And so then we can screen for any colonies that have picked up that recombinant DNA molecule, and then later on purify it out of the bacteria. We've essentially used the bacteria as a DNA copy machine to make many, many, many copies of our gene of interest. And then we can manipulate that gene of interest. We could mutate it to see what our mutation might do to the function of the protein product encoded by that gene. Lots of different things we could do. And we'll be covering quite a few of those later this week. So that gives you a flavor for Unit 1, defining the cell and its study. Once we've done that, once we've defined the structure function, we're going to dive into the cell and look at how information flow is regulated. And the best way to think about this unit is to conceptualize it over here with the cartoon. So here is a DNA molecule with a gene indicated. We know that transcription can occur where a promoter region within that DNA molecule could be bound by RNA polymerase, and then RNA polymerase could induce the transcription of that gene. What we'd end up with is this primary transcript. This is an mRNA molecule that includes exons and introns. Post-transcriptional processing would remove the exons, basically cut those out and religate the introns into the mature mRNA. That can get modified by polyadenylation, capping, maybe even rearrangement of some of the exons, and then it would leave the nucleus where all of these things have been happening, make it into the cytosol where it would then be translated by ribosomes, tRNAs, etc. Then we get the protein product. There it is. That's what's encoded by that gene. That's why we call proteins gene products. And then once that's made, we can modify it post-translationally. We can add things to it like sugar groups or phosphate groups from ATP. And then finally, after folding and moving, that protein is going to be targeted to its correct location within the cell. We're looking at how information in the flow of nucleotide bases in a gene flows through the cell up to the level of the protein. So Unit 2 is all about that information flow and how it's regulated. We're also going to link all of that information flow to the cell cycle. And you may remember that the cell cycle includes an M phase, mitosis, a G1 or growth 1 phase, followed by a synthesis phase, and followed by another growth G2 phase. So after cell division, we move, and that's what happens in mitosis obviously, we move into G1. We're essentially preparing for DNA replication. DNA replication occurs in S phase, and then in G2, we're really preparing to divide. So we're going to link information flow, which is over on this side of the screen, to the cell cycle as we move through this unit. Other things that we'll talk about. Some pretty cool newer discoveries involving microRNAs and small interfering RNAs, which are important regulators of gene expression and gene products. So we'll be giving you a feel for how the machinery of the cell that we covered in Unit 1 is used to regulate information flow. Finally, if you'll notice where we end up here, we end up with the protein. So now that we've learned how to build the proteins, our third unit is looking at protein pathways. Think of these as networks within the cell, built of, made up of these different proteins that do things like converting extracellular signals to intracellular work or change. So here's an example of what we would call a signal transduction pathway, and that's what I'm really indicating, or what I mean when I say protein pathway. So let's orient ourselves to the cartoon. Here's the lipid bilayer. This is the extracellular space. This is the cytosol. Outside the cell is a signaling molecule. We call these ligands. Signaling ligand binds to receptor in the membrane and induces a series of chemical reactions that activate something downstream. Think of this as protein X activating protein Y. And then protein Y can activate protein Z, and so on. We've got this existing network of pre-built proteins ready to respond to an extracellular signaling ligand to make something happen in the cell. Now in this cartoon, what's happening is mobilization of calcium and then downstream activation of calcium-dependent protein kinases, But often these networks are used to do things like regulate gene expression or the growth of a cell, a shape change. Lots of different things can occur. So let me recap. Unit 1, structure, function, and how the cell is studied. Unit 2, how information moves through the cell up to the level of protein. Unit 3, what happens to the proteins? That's the idea. So the main topics in Unit 3 include the idea of post-translational modification. Turns out a lot of these signaling networks are modified at the post-translational level after the protein's already been produced. How does the signaling work, and how are those mechanisms uncovered? And then some special topics as well. We'll do some primary literature introduction, and we'll do some work on stem cells and cloning as well as bioinformatics in that third unit. So we now come to the laboratory, a quick discussion of what we're going to be doing in the lab. Obviously, we won't be meeting in the lab. We won't be moving our hands to collect any data, but I have plenty of data to give you. And we will also, of course, talk a lot about the techniques and why we're doing them. So we'll virtually complete the laboratory. This laboratory is integrated into the content of the course. And throughout the class, you're going to see us refer to what is happening in the laboratory. That's intentional. The other thing that's intentional is the project-based organization of the lab. We're not going to do this as a series of modules. We're going to complete what we would consider a project. So the way that most of us organize our labs is we bring students into the lab, we teach them techniques, they practice those techniques, then they apply the technique to a hypothesis related to some aspect of our research. And what we're going to be doing here in the CELLAMELEC lab is essentially that. We're going to be looking at developing a set of techniques, at least virtually, and then applying those techniques to a particular hypothesis. And what we're going to be investigating in the lab is the competition between these two enzymes. They're called alpha and beta secretase. They're proteases. And we're going to be looking at how these enzymes compete for the cleavage of a particular substrate. And the substrate is called APP. APP is the protein substrate that the secretase enzymes cleave to produce something called amyloid beta. And this is a pathogenic fragment that leads to Alzheimer's disease. So we're studying the cell biology and the biochemistry of Alzheimer's disease in the lab. And I'll give you a lot more detail on what this is all about starting in week 2. Lab will not meet for week 1. Your first pre-lab lecture will occur on Thursday of week 2. Now, the way the laboratory will be integrated into the course for our online purposes is the following. On Thursdays, most Thursdays, I'll post by 11 a.m. a pre-lab lecture, and that will serve as the basis along with your lab manual for lab discussion questions, and those will be offered by Friday of the weeks that we're making them available, and due the following Wednesday, as we indicated before. Also, we'll be doing a final lab report. It will be brief. It will be essentially a regular lab report given our unique circumstances, and I'll give you a lot more detail on how to complete that as we move through the course. So conceptually, we're covering a particular project related to Alzheimer's disease, and sort of philosophically, we're trying to get you to transition from thinking of scientific investigation as a modular enterprise and thinking of it more as a holistic enterprise, where from week to week we're building upon things we've done previously to then build up to a big hypothesis test. Again, more on that logic and more about that during the pre-lab lectures. Finally, here I'm going to end part 1 with the sort of learning objectives for the course. Here they are. So first, I would expect that all of you obtain the knowledge that cells are central to the life sciences. They are the fundamental unit of life, And not surprisingly, they are central to all of the aspects of the investigation of the life sciences biology. Second, tool production and experimental approaches are key. We cannot generate new information without new technique and new tools. And of course, science is a process whereby we experiment and interpret to learn new knowledge, and we could not do that without our experimental approaches or our tools. Third, a lot of this relates to Unit 3 of the course. That's really what we're building up to. Post-transcriptional and post-translational control mechanisms really drive cell biology. We can't understand the structure, we can't understand the function without knowing those post-transcriptional and post-translational controls. And a couple of final points. One, macromolecular machines and changes in shape or conformational changes are essential to driving cellular function. I'll just give you one simple example. The ribosome. The ribosome is a complex macromolecular machine. In eukaryotes, it's made up of at least 5 rRNA molecules. It's at least 20-something proteins. And all of this is held together in a complex 3-dimensional structural unit. And if you took out any one of those component parts, the machine wouldn't work quite as well. And so we can think of these little proteins and RNA molecules as coming together to make a simple machine or a macromolecular machine. Now, the way that a lot of the machine mechanism works is through shape change, a change in the native or basic structure of the machine. So we call those conformational changes. That's something we'll talk a lot about in our course. Finally, the last thing I'll leave you with is a thought, philosophical idea. Dogma is constantly shifting. What I mean by that is, well, first let's talk about what dogma is. Dogma is, I like to think of it as the received wisdom of the elders. And this is the information that we use to write our textbooks and develop our lectures and design our courses. But we can't become entrenched in dogma because dogma is always changing. A good example is RNA. Since we're talking about RNA here, and rRNA in particular, did you know that the rRNA component of the ribosome is what's responsible for linking amino acids together via peptide bond formation? That's something we didn't realize until around the year 1998. And up until then, we would have assumed that it was the proteins in the ribosome that were important because most other enzymes are proteins. But as it turns out, RNAs can be enzymes too, and the rRNA component of the enzyme we call the ribozyme. Now, that discovery in the late 1990s was a huge paradigm shift in our thinking about RNA. No longer do we think of RNA as sort of the boring intermediary within the central dogma, which I'm writing out here. Now we think of RNA as much more than just a middleman. RNA is doing things like catalyzing covalent bond formation. So that's a shift in dogma, and so that's what I mean. You need to always respect the dogma. It helps us to organize our thinking, but never become too convinced or entrenched in that dogma because it will necessarily change. Okay, so that's enough for part 1. I'm going to take a short break, and then I will record part 2. Talk to you then.